And first, we're going to talk about California sea lions. And California sea lions is also known as Xolophus californianus. It's a coastal-eared seal native to western North America. And since our topic is it's going to talk about its behavior, so we are going to give you a little information about its mood behavior. The sea lions may travel alone or in groups, while at sea and how out between each sea trips. So what is how out? It means instead of staying in the sea waters, the sea lions will leave the water surface to land or sea ice. Introductions. And what's the background of our topic? With the rapidly changing of human-dominated environments, animals were shifting foraging strategies to explore these novel human-derived resources. And why do we study social associations? And social relationship defined as an individual position in social network, which can impact either spread of information or the behavioral variance. So by studying social network, it can allows us to know the social structure of the social transmission of foraging patterns. So why do we choose the place? In the Pacific Northwest of U.S., dam construction in salmon-bearing rivers create a novel concentration of solenoids. So the dam increase and the more concentration of the salmonoids. So we observe the data of the two sites where sea lions located the most along the Columbia River. And according to the data, we found that foraging in the Columbia River estuary is not new. However, observation of sea lions at the Bonneville Dam is rare. So we want to know why. So we want to know how an individual's centrality influences both discovery and the production success of the dam. So what is centrality? It means it helps us to know the importance of the individual among the network. So the purpose of this paper, California sea lions with population have grown in human-dominated environments might threaten or have an impact on salmon populations, and because they feed on them, so some of which are endangered. So we want to solve the problem, and we have to understand the problem first. So an individual population within a social network predicts discovery at a dam, so you want to know its effect. And second, we want to determine whether there are individual differences in predictions. And third, we want to know what effects influence why some individual becomes the repeat forager, means why do they keep coming back to the dam. And we have material and methods. So we have observation first. The first one is marine basin, which we call EMB. And California sea lions presence is well documented in the lower Columbia River, which is EMB. And we use side-reside data of the branded individual held out at the EMB and by the Pacific States Marine Fisheries Commission to create an association matrix. So what is association matrix? It's a statistics method to calculate association between pairs of individuals being seen together. And our second observation site is Bonneville Dam. And California sea lions predation on Solomon and Bonneville Dam has been monitored by the biologists. And they have recorded the number of the sea lion presents there by their brands and cards and scars like this, and the size and so on. And we want to determine which individuals from the EMB association matrix use two sites, the dam and the EMB. So we analyze. We elect to use the repeat and the co-occurrence, which means they show up together again and again and at the EMB to measure the social relationship over time. So as you can see, we have graphic picture, and you can see there are black nose and white nose. And black nose represent Bonneville foragers, and white nose represent non-Bonneville foragers. And as you can see, there are different sizes of black nose. The larger one means the sea lions capture more salmons. And then we want to know the social network positions. So we use 4 statistics, something like 4 social network centrality metrics. And it's kind of complicated, but I'm just surely describe it. The first one is betweenness. It's discover the path of information, how individual use it. And closeness centrality means the connectedness and the interaction between individuals. And like clustering coefficients means the social associations between groups. And eigenvalue centrality means how much an individual is connected to a much more connected individuals. So we want to know what is foraging success. So we define foraging success based on how much salmons the sea lion catch and fix a generalized linear mix, which is the applications, and with the total number of salmon predicted. With also these variations like the first observed arrival, or like call, which is a sort of the white animal, and the fourth central maxi, which is talked about. And we have our results. And on the EMB, we have the data for 1,007 sea lions being sighted. But I'm just surely show you guys because it's not really important, so just skip it. And we have our first questions. So whether individual use the Bonneville Dam foraging sites. And according to the data we observed, we have statistic numbers, and we found that the closeness here is positively associated with the use of Bonneville Dam, because it has a largest range of confidence interval and a smaller range of p-value. And we also found that eigenvalue centrality is negative associated with it. And our second question is about its prediction success based on how much salmon they predicted. And we found that the code presents here, and the days the sea lions stay at the dam is positive associated with the foraging success. And the mean daily conspicuous, which means same kinds of species, is negative associated with it. And we want to know whether the individuals return to the dam. And we found that the previous years, the sea lion stay and closeness once again is positive associated with the Bonneville return. However, the heterospecifics, which means different kinds of species, and conspicuous and betweenness is negative. And we have our discussions and conclusion. So from our observation, EMV may serve as an information center from which unsuccessful forager can follow successful forager to the dam, like this. So this is the EMV. And at the dam, prediction success was influenced by the individual foraging efforts. Means it's based on how much were they done, not because they're social factors we just mentioned about. I'll show you. Here, as you can see, the fourth centrality doesn't have a standout association with the foraging success according to the paper. And so it's more about how much they stay in the dam. And we also found that centrality like closeness are both positive, or as you can say, it's predict the discovery and return to the dam. And we also found that benefits of a foraging, if the benefits of foraging are high, high, and the effects of competition are magnified, and thus can cause our individual produce fewer salmon, and they probably won't want to come back anymore. And different centrality, like I just mentioned about, like this. Yeah, like, okay, that closeness here can predict differently like other centralities, because it might have to be due to different request application, like maybe for eigenvalue or clustering coefficient maybe need more complicated applications. And we also found that body size might be one factor influencing the foraging success. It may cause the bigger sea lions can have a better diving ability, but since our paper doesn't really talk about it, so we need more research about it. And a previous experience at a den doesn't explain foraging success, but naive individual interact with experienced bonobo foragers have better chance to success, which means the previous experience doesn't directly relate to the success at the dam, but they can follow like experienced foragers, and they have better chance to catch more salmon. So this is my talk. Thank you for listening.
The full social network metrics. Could you explain that again?
Okay. She wants to know more detail about the four centrality. So the first I'm going to talk about is betweenness, and it's about the like this here, the control path of the informations. So like this nodes, he has control more path about between in a network. And the closest centrality means how much interactions. So these two has more interaction and connectedness between the network. And the clustering coefficients is about the groups. So this is a group, this is a group, this is a group. So it's the connectedness and associates between groups. And the eigenvector centrality, we want to know how much an individual associate themselves connect to the others in the network. So this one has it's more connected to others, so his number is larger. And others, if they have more connected to this one, it's much more connected individuals, and they have higher numbers.
And can you explain again this picture, like a ball?
Okay, repeat. He want me to explain this picture. So this is E and B, and we like calculate the repeat co-occurrence, which means those sea lions show up together and get together, and we calculate and we analyze this, and we put it into the graph, and we have the black nose means they have show up in a den, and the white nose means they didn't show up in a den. And if they catch more salmons, the nose would be like larger, so this one might be like catch more salmons, and you can find that they are mainly focused on this area.
How can you get the 9.4 and 2.34 and 0.444? How can you get the three number?
You mean this, and this?
The 9.4 larger than 1 and
Do you mean this? Oh yeah. Yeah.
Yes,
Yeah.
yes.
He wants to know what's the number I show about here. So this is the one I like minus 10.7 to 1.3 to show you that it has a larger confidence interval. So this is also one. Like this one minus this one, and this is this one minus this one. So it has a smaller confidence interval. Yeah.
Okay, thank you.
What is the relationship between the four interaction pattern you mentioned and your research?
She wants to know what's the relationship between this four centrality between my paper. Yeah. Because they want to know the social network positions, so we use this for social network centrality to know the relationship between individuals and in different ways. And because they might have different results, but mainly discuss the interaction between the individuals.
Could you please explain what is CI? Yeah. What does CI mean?
Okay, she wants to know what is CI and P value. So CI is confidence interval, and it's a statistics methods. It's really complicated if you doesn't study statistics before, but it's like it's a range that whereas the star we analyze is more likely to be. Yeah. So just remember that this confidence interval is larger than something we analyze, like more likely to happen.
So how do we calculate the days at them of the sea lion?
She wants to know how we calculate the days at the sea lions stay at the dam. We didn't really calculate it. We use the there have been monitored by the United States Army Corporation and engineering biologists for decades, so we just use the data from them.
So you can see it's a very. So she gave a very fluent talk. Actually, I think she spoke too fast because this study is not that kind of study that talk about the weight of animal or length of animal, which are easy to comprehend. Instead, it talks about some abstract concepts like the the social network, which is also very difficult for me. So regarding these 4 parameters, so you have tried to explain during the QA session. But since it is the focus of this research, my suggestion is next time you can consider some analog for human society. That would be easier to comprehend. For example, my understanding is eigenvector would be someone like for example, like a bandai, right? This person has connection to everybody, but probably not that close. But he's the one to spread information to the whole class. That kind of analog. It would be easier to understand. Otherwise, after you explain all the 4 parameters, I already forgot the first one. So that's a problem we have because it's so abstract and they all look so close, so similar. Like betweenness, closeness, I don't know what's the difference. So you need to come up with some analog in human life. That would be easier to understand. And then some slides. And then because these 4 apparently key parameters, they are not that easy to understand or to remember what they really meant. So therefore, when you were presenting the results, you were like really, really fast. And it's not that easy for me to understand what the meaning really is. And then another thing is because you talk about all the results all together and really fast, and then you talk about the data. And then when you separate data and discussion, what you did during your talk is you always have to, or you do that 3 or 4 times, like you go backwards. That's something that we try to avoid in a very short talk because that takes time. So I have said several times that the best way to present your data is you talk about the data and then you immediately talk about its meaning so that we can have a straightforward connection between the data and its interpretation. Like this one is probably for the discussion of closeness. I don't know. I don't quite remember. So it tells us why some these forager keeps coming back, right? And that's related to this parameter. And then maybe that would be the place to show your discussion right after this. And as I mentioned also several times, try not to copy the table directly from the paper. Instead, try to, because it's a short table, try to remake the table. And in fact, if you don't want to go into the detail, you don't need to present the confidence interval because it's a little bit distracting. It's important for scientific paper, but in a short presentation, I would suggest that you can actually take away this. And you can only tell us the p-value. It's all fine. Yeah, so try to simplify the information by remaking the table. Yeah, and then And then some of the slides, I think it's a lot of information like this one. But I think you can keep it. Yeah, I just think it's a little bit too much information. And some of the fonts can be larger. Try to make the fonts as large as possible. I don't remember. Some of them are like this. I will try to make them larger whenever possible. Yeah. Like this. Oh, it's a little bit small to me. All right. But as you see that her presentation was very fluent, and she actually tried to present a relatively complicated ecology paper. So I think you will be able to better next time. Okay, thank you, Nina.